Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Spliceosome. 30 languages. Spliceosomal splicing cycle. A spliceosome is a large ribonucleoprotein, RNP, complex found primarily within the nucleus of eukaryotic cells. The spliceosome is assembled from small nuclear RNAs, snRNA, and numerous proteins. Small nuclear RNA, snRNA, molecules bind to specific proteins to form a small nuclear ribonucleoprotein complex, snRNP, pronounced SNRPs, which in turn combines with other snRNPs to form a large ribonucleoprotein complex called a spliceosome. The spliceosome removes introns from a transcribed premna, a type of primary transcript. This process is generally referred to as splicing. 1. An analogy is a film editor, who selectively cuts out irrelevant or incorrect material, equivalent to the introns, from the initial film and sends the cleaned-up version to the director for the final cut. Citation needed. However, sometimes the RNA within the intron acts as a ribozyme, splicing itself without the use of a spliceosome or protein enzymes. History, edit. See also, splicing, genetics. In 1977, work by the Sharp and Roberts labs revealed that genes of higher organisms are split or present in several distinct segments along the DNA molecule. 2. 3. The coding regions of the gene are separated by non-coding DNA that is not involved in protein expression. The split gene structure was found when adenoviral RNAs were hybridized to endonuclease cleavage fragments of single-stranded viral DNA. 2. It was observed that the RNAs of the mRNA DNA hybrids contained 5 and 3 tails of non-hydrogen bonded regions. When larger fragments of viral DNAs were used, forked structures of looped-out DNA were observed when hybridized to the viral RNAs. It was realized that the looped-out regions, the introns, are excised from the precursor RNAs in a process Sharp named splicing. The split gene structure was subsequently found to be common to most. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Eukaryotic genes. Philip Sharp and Richard J. Roberts were awarded the 1993 Nobel Prize in Physiology or Medicine for the discovery of introns and the splicing process. Citation needed. Composition, edit. Each spliceosome is composed of 5 small nuclear RNAs, snRNA, and a range of associated protein factors. When these small RNAs are combined with the protein factors, they make RNA protein complexes called snRNPs, small nuclear ribonucleoproteins, pronounced SNRPs. The SNRs that make up the major spliceosome are named U1, U2, U4, U5, and U6, so-called because they are rich in uridine, and participate in several RNA and RNA protein interactions. 1. The assembly of the spliceosome occurs on each premna, also known as heterogeneous nuclear RNA, hnRNA, at each exon-intron junction. The premna introns contain specific sequence elements that are recognized and utilized during spliceosome assembly. These include the 5' splice site, the branch point sequence, the polypyrimidine tract, and the 3' splice site. The spliceosome catalyzes the removal of introns, and the ligation of the flanking exons. Citation needed. Introns typically have a GU nucleotide sequence at the 5' splice site, and an AG at the 3' splice site. The 3' splice site can be further defined by a variable length of polypyrimidines, called the polypyrimidine tract, PPT, which serves the dual function of recruiting factors to the 3' splice site and possibly recruiting factors to the branch point sequence, BPS. The BPS contains the conserved adenosine required for the first step of splicing. Citation needed. Many proteins exhibit a zinc binding motif, which underscores the importance of zinc in the splicing mechanism. 4, 5, 6. The first molecular resolution reconstruction of U4/U6.U5 triple small nuclear ribonucleoprotein, tri-SNRNP, complex was reported in 2016. 7. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Figure 1. Above are electron microscopy, 8, fields of negatively stained yeast. Saccharomyces cerevisiae, tri-SNRNPS. Below left is a schematic illustration of the interaction of tri-SNRNP proteins with the U4/U6 snRNA duplex. Below right is a cartoon model of the yeast tri-SNRNP with shaded areas corresponding to U5, gray, U4/U6, orange, and the linker region, yellow. Creo-M has been applied extensively by Shi et al. To elucidate the near-forward/atomic structure of spliceosome in both yeast, 9, and humans. 10. The molecular framework of spliceosome at near atomic resolution demonstrates spp42 component of U5 snRNP forms a central scaffold and anchors the catalytic center in yeast. The atomic structure of the human spliceosome illustrates the Step 2 component SLU7 adopts an extended structure, poised for selection of the 3' splice site. All 5 metals, assigned as Mg2+, in the yeast complex are preserved in the human complex. Citation needed. Alternative splicing, edit. Main article, Alternative splicing. Alternative splicing, the recombination of different exons, is a major source of genetic diversity in eukaryotes. Splice variants have been used to account for the relatively small number of protein coding genes in the human genome, currently estimated at around 20,000. One particular Drosophila gene, PSCM, has been speculated to be alternatively spliced into 38,000 different mNAs, assuming all of its exons can splice independently of each other. 11. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Location of splicing, edit. Premna splicing factors were originally found to be concentrated in nuclear bodies known as nuclear speckles. 12. It was originally postulated that nuclear speckles are either sites of mRNA splicing or storage sites of mRNA splicing factors. It is now understood that nuclear speckles help concentrate splicing factors near genes that are physically located close to them. Genes located farther from speckles can still be transcribed and spliced, but their splicing is less efficient compared to those closer to speckles. 13. RNA splicing is a biochemical reaction, and like all biochemical reactions, its rate depends on the concentration of enzymes and substrates. In this case, the enzymes are the spliceosomes, and the substrates are the preimners. By varying the concentration of spliceosomes and preimners based on their proximity to nuclear speckles, cells could potentially regulate the efficiency of splicing. 13. Assembly, edit. The model for formation of the spliceosome active site involves an ordered, stepwise assembly of discrete snRNP particles on the preimner substrate. The first recognition of preimners involves U1 snRNP binding to the 5' splice site of the preimner and other non-snRNP associated factors to form the commitment complex, or early E complex in mammals. 14, 15. The commitment complex is an ATP-independent complex that commits the preimner to the splicing pathway. 16. U2 snRNP is recruited to the branch region through interactions with the E-complex component U2F. U2 snRNP auxiliary factor, and possibly U1 snRNP. In an ATP-dependent reaction, U2 snRNP becomes tightly associated with the branch point sequence, BPS, to form complex A. A duplex formed between U2 snRNP and the preimner branch region bulges out the branch adenosine, specifying it as the nucleophile for the first transesterification. 17. The presence of a pseudoridine residue in U2 snRNA, nearly opposite of the branch site, results in an altered conformation of the RNA-RNA duplex upon the U2 snRNP binding. Specifically, the altered structure of the duplex induced by the pseudoridine places the 2O of the bulged adenosine in a favorable position for the first step of splicing. 18. The U4/U5/U6 tree snRNP, c. Figure 1, is recruited to the assembling spliceosome to form complex B, and following several rearrangements, complex C is activated for catalysis. 19, 20. It is unclear how the tree snRNP is recruited to complex A, but this process may be mediated through protein-protein interactions and forward/or base pairing interactions between U2 snRNA and U6 snRNA. Citation needed. The U5 snRNP interacts with sequences at the 5 and 3 splice sites via the invariant loop of U5 snRNA. 21. And U5 protein components interact with the 3 splice site region. 22. Upon recruitment of the tree snRNP, several RNA-RNA rearrangements precede the first catalytic step and further rearrangements occur in the catalytically active spliceosome. Several of the RNA. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. RNA interactions are mutually exclusive, however, it is not known what triggers these interactions, nor the order of these rearrangements. The first rearrangement is probably the displacement of U1 snRNP from the 5 splice site and formation of a U6 snRNA interaction. It is known that U1 snRNP is only weakly associated with fully formed spliceosomes. 23, and U1 snRNP is inhibitory to the formation of a U6-5 splice site interaction on a model of substrate oligonucleotide containing a short 5 exon and 5 splice site. 24. Binding of U2 snRNP to the branch point sequence, BPS, is one example of an RNA-RNA interaction displacing a protein-RNA interaction. Upon recruitment of U2 snRNP, the branch-binding protein SF1 in the commitment complex is displaced since the binding Site of U2 snRNA and SF1 are mutually exclusive events. Citation needed. Within the U2 snRNA, there are other mutually exclusive rearrangements that occur between competing conformations. For example, in the active form, stem loop IA is favored; in the inactive form, a mutually exclusive interaction between the loop and a downstream sequence predominates. 20. It is unclear how U4 is displaced from U6 snRNA, although RNA has been implicated in spliceosome assembly, and may function to unwind U4/U6 and promote the formation of a U2/U6 snRNA interaction. The interactions of U4/U6 stem loops I and II dissociate, and the freed stem loop 2 region of U6 folds on itself to form an intramolecular stem loop, and U4 is no longer required in further spliceosome assembly. The freed stem loop I region of U6 base pairs with U2 snRNA, forming the U2/U6 helix I. However, the helix I structure is mutually exclusive with the 3/2 of an internal 5-stem loop region of U2 snRNA. Citation needed. Spliceosome, edit. See also, Spliceosome. Some eukaryotes have a second spliceosome, the so-called Spliceosome. 25. A group of less abundant SNRs, U11, U12, U4 attic, and U6 attic, together with U5, are subunits of the minor spliceosome. That splice is a rare class of premner introns, denoted U12 type. The minor spliceosome is located in the nucleus like its major counterpart, 26, though there are exceptions in some specialized cells including anucleate platelets, 27, and the dendroplasm, dendrite cytoplasm of neuronal cells. 28. References, edit. 1 AB Will CL, Lumen July 2011. Spliceosome Structure and Function. Cold Spring Harbor. Perspectives in Biology. 3. 70037007 do 10.1111/perspect.0037007 PMC 3119917 PMID 2144158 2 AB Burgett SM, Morsi, Sharp Pa, August 1977. Spliced segments at the 5 terminus of adenovirus. Too late mRNA. Proceedings of the National Academy of Sciences of the United States of America. 74. 83171-5. Bibcode, 1977 NAS, 74.3171b. Do. 10.1073/NAS.74.8.3171. PMC 431482. PMID 269380. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 3 Chow LT, Roberts JM, Lewis JB, Broca TR, August 1977. A map of cytoplasmic RNA transcripts from lytic adenovirus type 2, determined by electron microscopy of RNA-DNA hybrids. Cell 11, 4. 81936. Do. 10.1016/0092-8674, 77, 90294x. PMID 890,740. S2 SID 379671144. 4 Agafonofte, Deckert J, Wolfie, Odinwalder P, Bissenhoff S, Will C. L, It al. July 2011. Semi-quantitative proteomic analysis of the human spliceosome via a novel two-dimensional gel. Electrophoresis method. Molecular and cellular biology. 31, 13 2667 82 do. 10.1128/MCB.05266. 11. PMC 313332. PMID 2153665. 5 Kuhnen, Van Santen Ma, Schweenhorster, Alorbach, Aluminar, January 2009. Stalling of spliceosome assembly at distinct stages by small molecule inhibitors of protein acetylation and destylation. RNA. 15, 115375 do. 10.1261/RNA.1332609. PMC 2612777. PMID 1902938. 6 Patil V, Canzoneri JC, Samatov TR, Aluminar, Euleri AK, September 2012. Molecular architecture of zinc chelating small molecules that inhibit spliceosome assembly at an early stage. RNA. 18, 91605 do. 10.1261/RNA.034819.112. PMC 34257776. PMID 228322025. 7 Kate JH, March 2016. Structure. A Big Bang in Spliceosome Structural Biology. Science. 351. 6281,392 do. 10.1126/science.arf4465. PMID 270137112. S2 SID 2066481885. 8 Hacker I, Sander B, Gullers mm, Wolfie, Karagoz, Kastner B, It al. November 2008. Localization of PRP8, BR2, SNU114 and U4/U6 proteins in the yeast tree SNRNP by electron microscopy. Nature. Structural and Molecular Biology. 15, 11 1206 do. 10.1038/NSMB.1506. PMID 1895335. S2 SID 22982227. 9 Yang C, Hang J, Wang R, Huang M, Wang CC, Shi Y, September 2015. Structure of a yeast. Spliceosome at 3.6 angstrom resolution. Science. 349, 6253 1182 91. Bibcode, 2015 SCI, 349.1182Y. Do. 10.1126/science.aac7629. PMID 262927007. S2 SID 22194712. 10 Zhang X, Yang C, Hang J, Finsai Li, Lei J, Shi Y, May 2017. Anatomic structure of the human Spliceosome. Cell 169, 591892.9.E14 do 10.1016/j.cell.2017.04.033 PMID 285027770 11 Schmucker D, Clemens J.C., Shu H., Werby C.A., Zial J., Muda M., It al. June 2000. Drosophila. Pscomyzin axon guidance receptor exhibiting extraordinary molecular diversity. Cell 101, 6671. 84 do 10.1016/S0092-8674, 00808778-8. PMID 10892653. 12 Spectre DL, Lamande I, February 2011. Nuclear speckles. Review. Cold Spring Harbor Perspectives. In Biology. 3, 2000646 10.1101/perspect.0000646. PMC 3039535. PMID 209265517. 13 AB but P, Chao, Emert B, It al. May 2024. Genome organization around nuclear speckles. Drives mRNA splicing efficiency. Nature. 629, 1165 1173 do 10.1038/S41586-024-07429-6. PMC 11164319. PMID 3872076. 14 Jamison SF, Crowe, Garcia Blanco Ma, October 1992. The Spliceosome assembly pathway in mammalian extracts. Molecular and cellular biology. 12, 10.4279/87. Do 10.1128/MCB.12.10.4279. PMC 360351. PMID 13836887. 15 Srafin B, Rosbach, October 1989. Identification of functional U1-SNA complexes. Committed to Spliceosome assembly and splicing. Cell 59, 234958 do 10.1016/0092. 8674, 89, 90296-1. PMID 2529976. S2 SID 18553973. 16 Legrain P, Srafin B, Rosbach M, September 1988. Early commitment of yeast Premna to the Spliceosome pathway. Molecular and cellular biology. 8, 9 3755 60 do 10.1128/MCB.8.9.3755. PMC 365433. PMID 3065622. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 17 Query CC, Moore MJ, Sharp Pa, March 1994. Branch nucleophile selection in Premna splicing. Evidence for the bulged duplex model. Genes and development. 8, 558797. Do 10.1101/gad.8.5.587. PMID 79266752. 18 Newbie Me, Greenbaum NL, December 2002. Sculpting of the spliceosomal branch site recognition. Motif by a conserved pseudoridin. Nature Structural Biology. 9, 12/95865 do 10.1038/NSB873. PMID 124266583. S2 SID 396286664. 19 Burge CB, Tuschl T, Sharp Pa, 1999. Splicing precursors to MNAs by the spliceosomes. In Gashtel RF, Sec TR, Atkins JF, eds. The RNA World. Cold Spring Harbor Lab. Press. pp. 52560. ISBN 978-0-87969-380-0. 20 AB Staley JP, Guthrie C., February 1998. Mechanical devices of the spliceosome, motors, clocks, springs, and things. Cell. 92, 331526 do. 10.1016/S0092-8674, 00809255-3. PMID 9476892. 21 Newman Tijl Campes, Beggs J.D., November 1995. snRNA interactions at 5 and 3 splice sites. Monitored by photoactivated cross-linking in yeast spliceosomes. RNA 1, 996880. PMC 1369345. PMID 8548661. Archived from the original on 23 February 2005. Retrieved 7 March 2008. 22 Chiara MD, Palangian L, Feldkramer R, Reed R, August 1997. Evidence that U5-snRNP recognizes the 3-splice site for catalytic step 2 in mammals. The EMBO Journal. 16, 15 4746-59. Do 10.1093/embodge/16.15.4746. PMC 11700101. PMID 9303319. 23 Moore MJ, Sharp Pa, September 1993. Evidence for 2 active sites in the spliceosome provided by stereochemistry of Premna splicing. Nature. 365, 6444 3648. Bibcode, 1993 Nature.365364m. Do, 10.1038/3653640. PMID 8397340. S2 SID 4361512. 24 Con40 BB, Kazuyoki MJ, Kanaska mm, December 1993. Disruption of base pairing between the 5-splice site and the 5' end of U1 snRNA is required for spliceosome assembly. Cell. 75, 5863. 73 do 10.1016/0092-8674, 93, 90531T. PMID 8252623. 25 Patel A. Stitz J.A., December 2003. Splicing Double, Insights from the Second Spliceosome. Nature Reviews. Molecular Cell Biology. 4, 12/9/60/70 10.1038 NRM 1259. PMID 1468517 S2 SID 218169110. 26 Pesa HK, Will CL, Meng X, Schneider C, Watkins NJ, Perala N, it al. June 2008. Minor. Spliceosome components are predominantly localized in the nucleus. Proceedings of the National Academy of Sciences of the United States of America. 105, 25 8655 60. Do. 10.1073/NAS.08036466105. PMC 2438382. PMID 1855950. 27 Dennis mm, Tolley ND, Bunting M, Schwartz H, Jiang H, Lindemann S, it al. August 2005. Escaping the nuclear confines, signal-dependent Premner splicing in anucleate platelets. Cell 122, 3379. 91 Do. 10.1016/J.Cell.2005.06.015. PMC 44019993. PMID 160960558. 28 Glantzer J, Miyashio KY, Seoul JY, Barrett L, Belt B, Haydon P, it al. November 2005. RNA splicing. Capability of live neuronal dendrites. Proceedings of the National Academy of Sciences of the United States of America. 102, 46 1685964. Bib code 2005 NAS 102168559G. Do. 10.1073/NAS.05037833102. PMC 1277967. PMID 1627597. Further reading, edit. Butcher S.E., 2011. Chapter 8. The Spliceosome and its metal ions. In Sigil Sigil H, Sigil RK, eds. Structural and catalytic roles of metal ions in RNA. Metal Ions in Life Sciences. Volume 9. RSC Publishing. pp. 23551. Do. 10.1037894184973251200235. ISBN 978-1-84973-094-5. PMID 2201024. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Nilsson T.W., December 2003. The Spliceosome, the most complex macromolecular machine in the cell. Bioessays. 25, 12 1147 9 Do. 10.1002/BIS.10394. PMID 146352448. External links, edit. 3D macromolecular structures of spliceosomes from the M Data Bank, EMDB. Spliceosomes at the U.S. National Library of Medicine. Medical subject headings, MESH. Categories: Spliceosome organelles, gene expression, protein complexes, RNA splicing. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.